Okay, let's go ahead and get started. Today we're doing a thesis defense, and most of you know the drill. But actually, before we do that, a couple announcements. And this is the last time you'll hear this from me, but next Wednesday night is a great movie. And I know I think many, if not most of you are signed up. Kevin Johnson, who's the chair of biomedical informatics at Vanderbilt, and he's actually on Thursday talking in this conference and then meeting with the fellows. And it's just kind of open. It's something that I always enjoy doing when I go to other places. And we can talk a little bit before then, but if you guys, however you guys want to organize, either present some of your ideas to him, you get his feedback, ask him if he's hiring, etc. Okay, then one other event I'm going to start mentioning is on Friday, October 28th, is the celebration of the 25th anniversary of the BIC building. We have tentatively lined up, and she has tentatively agreed, pending her starting her new job. The other email you got from me yesterday, that Dr. Patty Brennan has been named the director of the National Library of Medicine, which I think is good. I think most people think is good. And anyways, we had always intended to invite the new director to that. And after I sent my congratulatory email, I said, by the way, I'm inviting you to come visit OHSU on October 28th, and she will be coming. Well, she tentatively said she will be coming. She actually doesn't start the job until August, although those of you going to the NLM meeting will get to meet her in June in Columbus. Can't remember the last time she was here, but she has a very high presence at AMIA. A number of you already know her. She's a really great person, a great choice for the job. And also at the event on the 28th will be Dr. Don Lindberg, the former director for 30 years. He will be here too, and a bunch of others. So it should be a fun day. And there will probably be opportunities for you guys to, I don't know, put up posters or something like that. We're still planning the day. Anyways, let's move on to our session today. As I mentioned, it's a master's thesis proposal, so many of you have been through that, where the process is that we go through the student who's defending his or her thesis, gives a presentation. There's then public questions, so anyone in the audience can participate. Then we all leave, leaving behind just the committee, or whoever is here of the committee. And then they can ask him further questions. Then he's dismissed, and they decide whether or not he passes. And if he passes, he's done. Although he's not totally done, he still actually needs to write up his thesis. Even if he, are you marching in the graduation, or okay, yeah, because sometimes people march in the graduation, usually at the anticipation that they've actually completed everything. But anyways, so I don't want to take too much of Ben's time, but let me just introduce him briefly. He received his bachelor's degree in psychology with an emphasis in neuropsychology and minor in political science from Whittier College. After graduating, worked as a research technician at University of California, Irvine, in aspects of neuroscience. And he then, like many of you, left the wet lab for drier pursuits, and did a number of things, including web applications and data visualizations, and ended up here. And so he has been pursuing the master's degree, and today we're going to hear the results of his work. So take it away. I'm sorry, before you go further, I goofed. I thought you were actually defending your thesis. No, you're defending your proposal. Gotcha.
That's correct. It's the proposal.
Gotcha.
See if it's okay.
Let's It's not the first time I made that mistake.
So to begin, sorry, it's kind of loud. I'll begin by introducing my topic and discussing the mutational landscape of cancer, and specifically the variants of interest to this research, how we detect them, and the sample properties that influence their detection. From there, I'm going to provide a background on the problem space and how we model background error in sequencing data, and I'll be discussing the specifics of frequently encountered types of background error, current approaches we use to modeling it, and then discuss my research question and hypothesis. From there, I'll introduce my specific aims, the methods I plan on using, the data I'll be working with, and the potential impact of this research, finally by a Q&A, or followed by a Q&A. So, as many of you undoubtedly know, variants come in a wide variety of physical structures, which are visible through the comparison of a sample or a sequence to a reference genome. And some of the most basic types of variants are the single nucleotide variants, insertions, deletions, mutations. And in addition to these, we have the longer substitutions, deletions, insertions, inversions, duplications. And these, in this slide, are kind of described as just structural variants. And of course, while not explicitly pictured here, we also have chromosomal variants, which are similar and can infect the chromosome or an entire arm length of the chromosome. For the purpose of this research, I'm going to be focusing solely on single nucleotide variants. So in addition to the structural distinction, they come in two forms, somatic and germline. And to the right, we see germline variants, and these arise from germ cells and are often discussed in the context of their heritability from a parent individual to an offspring. Generally speaking, when these variants are inherited, they are present in the DNA of all the cells of the body. In contrast, somatic variants, which are to the left, differ in that they arise spontaneously within the cells throughout the lifetime. And these are the mutations that are more often associated with cancer, among other disorders that are kind of resulting from tissue-specific dysregulation. Throughout this presentation, when I talk about variants, I'm only going to be really referring to the somatic variants. Otherwise, I'll specify that I'm talking about germline. So this may take a second to load. Oh, there we go. So for single nucleotide variants, their detection comes down to allele frequency, which in this context can be described as the proportion of a population of cells in a sample that exhibit a given variant allele. So in this kind of abstract representation, we see allele frequencies on the y-axis, and we see the physical base position on the x-axis. And I'll note that these are not genomic coordinates. My apologies. So if we break these out by kind of their dinucleotides and imagine that we've been charged with the confident detection of single nucleotide variants using this graph and only our eyes, we might pick out these 4 alleles on the left. And then we'd report that we detected 4 single nucleotide variants in the target sequence. And the rationale behind this choice may be that the data points on the graph, these are probably variants. And we pick them because they appear to have a significant deviation from the background variation in allele frequency. And we may argue that this deviation indicates that there is, in fact, a proportion of cells in the sequence population that truly exhibits these variants. But if we look closer, we can also discern that there looks to be some background error and noise. And that is representative there. And then when we kind of see the background error and noise, we say, okay, well, there's also this region here, and maybe we'll call that a low confidence region. That's an area where there seems to be a significant deviation, but we can't really discern whether it's meaningful or not. And so when we consider that, we may say, okay, well then this is our high confidence threshold. Above here, we can be confident in the detection of variants. Below, we're not actually so sure. So when we keep this in mind and we're discussing our power to detect variants from sequencing data, we're often referring to minor allele frequency. Simply put, minor allele frequency is the frequency at which the least common allele occurs in a population of interest. So for the example of visually detecting variants using this figure, we may say for simplicity that our detection limit is about 0.14 or 1.4% NAF, meaning that we can detect with high confidence any alleles present in that population existing at a frequency higher than 1.4%. So of course, if detecting variants was that simple, this presentation probably wouldn't be happening. Allele frequency is heavily influenced by a number of sample properties, which include purity, clonality, and copy number. The major sources, and these are major sources of confounding in a sample. When these dynamics combine, the frequencies of variant alleles in a sample are quickly confounded and often reduced, making their confident detection difficult. So from this research, I'll primarily be focusing on purity and clonality. Copy number is a little harder to get traction on and probably would be more for a dissertation. Now, before getting into the specifics of sample purity, I should emphasize that whenever we sequence a biological sample, more often than not, and I kind of hinted at this earlier, we're sequencing a population of cells. This is a critical point because the cell population contains variant subpopulations. And when we sequence the population of that sample, the proportion of sequence reads that exhibit that
You
variant.
are the only participant in the conference. Please press any key on your keypad to remain in conference.
Womp womp. The proportion of sequence reads that exhibit this variant should be roughly proportion or equivalent to the allele frequency in the entire population. And this is kind of the ideal scenario. So that said, when here we have an abstract representation of a blood vessel with peripheral blood. And if we draw blood and sample it, we're going to be getting a sample of that population. And it's important to note that we're going to get normal cells in there too, which reduces the purity of the sample and also reduces the allele frequencies. And this can be kind of a confounder. So before assessing and calling variants, this is something we seriously need to...
You are alone in the conference. Disconnect.
All right. Okay. Cool. Load. So yeah, this reduced sample purity is a confounder that we really need to account for in any modeling of error. So in addition to purity, there's also tumor clonality, and this is kind of a handful. The idea is that among a tumor, there's heterogeneous clonal populations with multiple genetically distinct subclones. And these subclones can each house different behaviors and exhibit different characteristics that kind of, when you combine them, attribute or generate the phenotypic characteristics of a given cancer. To understand how clonality develops, it helps to first understand how cancer develops. The first step on the path to cancer is the initialization of oncogenesis with an initial driver mutation. After this initialization, we also often see a clonal expansion where malignant cells with the initial driver mutation pick up additional mutations. So at this point, if we were to sequence a sample of this malignancy, we would see multiple subclones, likely with a common driver mutation, and different variable variant allele frequencies for other mutations. And maybe this would be the time that you would get diagnosed and start your first round of treatment. Of course, as we all know, oftentimes treatment isn't completely effective. And while the population of subclones may be reduced significantly, we'll probably have also selected for the resistant subclones in this population. And this is really classic population genetics. At this point, if we assume a treatment response, the variant allele frequencies will have been reduced due to this reduction in malignant cells, but we'll also have had that population shifted. Now, if we imagine that because of these surviving subclones, we were to relapse once again, we may choose to receive another round of treatment. And we'll see a similar situation, potentially, where disease burden and variant allele frequency is reduced from the treatment, but we're also getting a selection of these malignant subclones and getting a shifting clonal structure or population structure. So in the context of detecting cancer cells and by their single nucleotide variant markers, the challenge really increases with treatment success, which is a little bit ironic. I mean, for the further challenges in, further challenges in the analysis, these variants are also compounded by the shifting structure of the population. So, altogether, in the narrowed context of single nucleotide variants, the mutational landscape of cancer exhibits a significant level of base complexity. And this really comes down, in this case, to the purity of the sample and the structure, the clonal dynamics of the population. So next I'll be covering some background. Shifting gears, the way we detect these variants is through complex computational pipelines that apply a statistical framework. Using these pipelines in next-generation sequencing, the power to detect variants is largely contingent on the read depth of the data. That said, sequence coverage also plays a significant role, because if we kind of miss the variant entirely, we're not going to find it. So if we keep this in mind, we can approach the problem in a statistical framework as such. With a lack of coverage, which we see on the bottom, we're just going to miss our variants. If we have correct coverage but inadequate depth, we're going to detect variants. We're not going to be necessarily very confident in them, and we also may discard variants for background area. Now, if we are adequately powered and have coverage, we'll be getting high confidence variants, and we'll also be separating out the platform error. But we'll still have these low confidence variants, and those are the ones we're really interested in. So, and it should be noted that currently studies really are sitting up on top. We do have adequate depth, and coverage is really down to experimental design. If the experimental design didn't work, that's when coverage would be an issue. So at present, the dominant pipelines for variant calling are GATK, or the Genome Analysis Toolkit, and MuTect, which are both from the Broad Institute. And GATK focuses on germline variants, while MuTect focuses on somatic variants. And these pipelines are often used in conjunction with the Illumina sequencing technology. And GATK in particular has seen a lot of success in genome-wide association studies. So as I mentioned, across major pipelines, there's the suspect in the case of the missing variants is likely hiding in the pre-processing steps, which here would mean BQSR, which is the background error model for GATK and MuTect. And the way BQSR works is it kind of takes in a lot of platform error and machine-specific context, and it uses that to update quality scores on the actual sequence so that the downstream variant caller can have better data to work with and call these variants more effectively. And of course, what is a quality score? Well, if you imagine that along each base we're performing a hypothesis test, the quality score is going to be kind of like a p-value. It's going to give us a level of understanding of how confident we can be in a given base call. So relevant to this research, there are a number of potential weaknesses in some of these error models, not just BQSR, in the context of calling low-frequency variants. It should be noted that a lot of these pipelines were established to build high confidence around variants and not surface low-confidence variants. So I'll touch on these in a bit, but so before I go forward, I'd like to discuss some basic platform errors. So I'll get to that next. So the first error is GC content bias, and this refers to an increase in GC DNA base content in sequencing reads. If we look at the figure here, we see that GC content covaries significantly with sequence coverage. On the y-axis, we have the coverage along a sequence by 1,000 base pair windows, and by the percent of GC content on the x-axis. So as we can see, as coverage decreases, we observe significant bias towards G and C bases in the sequence. We also see that coverage is heterogeneous, with the vast majority of windows having coverage below 6,000 reads. This additional heterogeneity can complicate the assessment of GC content within sequencing data. In addition to that, we also have homopolymers. Homopolymers relate to repeating base sequences in the DNA. Machines, a lot of the platforms have trouble delineating or assessing these regions. And we can see that they're having trouble when we align them to the reference and see that error rates as we match these. So that's just the basics. It gets even more complex. In the top left of the slide, we see that base insertions, deletions, and substitution error rates are also by homopolymers. And in fact, we also see the same for GC content line. And we can also notice that this is species specific. So color saturation rate will be relative. That really kind of factors in this very index species. And then of course, it varies by platform. All the platforms have different error profiles, and this really complicates things quite a lot. And so maybe the simple solution could be, let's do research on the individual error platforms, let's generate error profiles, and then essentially curate a library of these profiles for each of these technologies and their species. But that would quickly generate a pretty complex library, and it's not really a scalable solution. So what researchers were focusing on is developing error models that actually do this, I'm going to go over some classical statistics stuff. It's, they use distributions, empirical and theoretical distributions. And empirical distributions, as many of you may know, leverage this observed data. And these distributions may not be ideal because they exhibit significant bias, or have the potential to exhibit significant bias towards the available data. And while not necessarily bad, these distributions can make reproducibility and cross-model comparisons a little more challenging. Theoretical distributions, on the other hand, these are distributions such as Gaussian and Bernoulli distributions. These are the ones we may be most well acquainted with in our statistics classes, and these are a little more robust to analysis and have much better understood assumptions. And with these with an understanding of these assumptions, we can actually do such as Gaussian distributions, different types of clustering. So in addition to these distributions, background error models also frequently use thresholding. And this is done to distinguish signals of interest from background error. These thresholds generally come in two flavors, and there are global thresholds, and these essentially are similar to a statistical test. In the case of quality scores, imagine aligning this distribution. So if you imagine aligning these distributions in sequence, we would essentially accept bases as being significant when they kind of reach the tail end of the distribution at a threshold set. In contrast, when we look at local distributions, what we're trying to do is really use sequence-specific context as we move across the distribution to better inform how we're calling these or detecting these true signals. And so we may use a sliding windowed approach or a K-mer approach, or just use an entire read. And if we imagine that, the distribution's moving along, we could also see it kind of shifting up and down. And that really is kind of the key to this. So in addition to distributions and thresholds, one of the common techniques we see too is clustering. And generally speaking, the common goal of clustering is to generate an error in error models is to generate sets or clusters of trusted sequences and reads using some heuristic. And once we have a trusted set, we can take the less trusted sequences and kind of correct them towards these trusted sequences, also using an heuristic approach. For the first, on the left, the K-mer spectrum approach, we often cluster by an edit distance measure, and that's usually something like Hamming distance, which is the minimum number of changes that can be made to a sequence to match the reference, without considering insertions and deletions. Or the Levenshtein distance, which is similar but does consider insertions and deletions. Another approach, which we see to the right, is to cluster based on a k-mer frequency. And this assumes that the more frequent a k-mer is, the more we can trust it. And of course, with a k of 3, this wouldn't be very informative. And so usually k will be much larger. And that kind of gets to how do we choose k, and a little out of scope, but it's kind of its own art form. So another approach very similar to clustering is to use graph models. And we would cluster by frequency, excuse me. And these are very similar to clustering by frequency. And they construct k-mer graphs, which are a common approach taken by sequence assembly software to do error correction during the assembly process. One example which I have here is de Bruijn graphs. And in graph theory, de Bruijn graphs are n-dimensional directed graphs of a set of symbols m that overlap one another. So what does that mean? In the context of error correction, a k value is selected to break sequences into overlapping k-mer nodes, and where the overlaps represent the edges of the de Bruijn graph. From this, sequence assembly software is able to assemble the reads into a consensus sequence, which we often call a contig, and sequences that and correct sequences that are poor matches to their closest k-mer. While de Bruijn graphs are just one example, there are actually many other graphing techniques that are also used across error models. And so, in considering these varied approaches, it's helpful to kind of use them as kind of features to break down these error models and kind of understand what error models are using currently. Here I've done just that for a very small subset of these models, and these models are not necessarily standalone. They may be found in existing software. From this, we see that the landscape with each method, it's very diverse, and each of these models has its merits and may have its drawbacks too, which often surround the context for its use. We see empirical thresholds and theoretical thresholds, and down here we see a combination of the two, and this is really kind of leveraging the power of both. Similarly, we also see, that was distributions. We also see different global and local thresholds, and locals can be based on haplotypes, windows, and often combining these, you can get a better estimation of the variation of the sequence. They also use very different correction methods. Some use k-mer frequencies in clustering. Others use kind of mixture models, and others use expectation maximization. So there's a lot of different approaches here, and each kind of has their own perks and potential drawbacks. And of course, the errors accounted for aren't necessarily all the same. Some really focus on detecting GC content and correcting for that, while others may be more interested in homopolymers or indels. So with that, my research question is our power to detect low-frequency variation being reduced by current background error models in our pipeline? And with that, kind of turn to the hypothesis, which is that we can improve the detection of low-frequency variance significantly through the development of a novel background error model that is actually optimized for this specific context. And with that, my first aim will be to create a framework that evaluates current background error models and leverages these evaluations for the development of an enhanced or novel background error model that is designed specifically for this use case. And then the second one will be to apply the final background error model to the task of detecting minimum residual disease in leukemia, which is a particularly challenging kind of use case and be a great kind of motivational example. So the evaluation framework will seek to evaluate the error models using GATK and MuTect as the test bed, and their application and they'll be applied to detect low frequency variance under multiple conditions. The first condition will be to run GATK and MuTect just without BQSR, their native background error model. And this will be to kind of establish, okay, is BQSR actually helping us detect these low frequency variants? And from there, we'll iterate through and essentially adapt additional error models into this pipeline, not necessarily in place of BQSR, but at their most rational point in the pipeline, and gather statistics and try and really understand how these error models are performing. And once we've got these statistics, we can use them to kind of rank these and see which ones are really doing best for the context we're interested in. So performance is going to be assessed in pretty standard sensitivity, specificity, precision, and accuracy. And these really get at what we're seeking, especially we're going to want to have high sensitivity with low frequency and hopefully also high specificity. So in addition to these standard statistics, also be using precision recall curves to visually assess the trade-off in precision and recall between models. And this will be a nice way to really understand the data. The first set of data I'll be using is whole genome sequencing synthetic data generated by the ICGC-TCGA Dream Challenge. And this data is essentially real human data that has been heavily modified using a software called Vantage. So we have a truth set. We know what variants should be in this data. And it also exhibits variable purity and variable subclone variant. So this is really ideal. Exactly what we'd be looking for. It is lower depth than what we are interested in, but to account for that, we'll also be doing a fixed dilution series, or the Night Diagnostic Lab will be doing one. And this will take normal and tumor sample pairs. And the tumor samples generated from will be generated with 80 to 90% purity and variable allele frequencies. And this will essentially be a known truth set too. That's more relevant to the application we're actually seeking to apply this model to. These samples will be sequenced using a 43-gene leukemia panel that's used at OHSU, and or previously was used at OHSU, and this was done on the Ion Torrent PGM platform. And the specifics of this data are still being discussed, as you probably can see from the slide. So for my second aim, I'll be seeking to apply this novel background error model to the challenge of detecting minimum residual disease. And so you may be wondering, what is minimum residual disease? Minimum residual disease is defined as the post-therapy disease burden. And this is particularly considered in the space of cancer and hematologic malignancies. So minimum residual disease, kind of a little background on what they can do for us. They provide really valuable feedback to oncologists and allow them the opportunity to kind of track disease and therapy progression. And in certain cases, they can predict relapse, pursue a more agile treatment plan, and avoid unnecessary drug toxicity or hematopoietic stem cell transplants. Altogether, these advantages can translate into really meaningful and tangible benefits to patient care and outcomes. So if we consider this, high confidence detection of minimum residual disease is really valuable. Unfortunately, given the nature of success when treating, which I noted when we were discussing clonality, monitoring minimum residual disease gets more challenging as our treatment progresses and those clonal populations kind of contract and lose their frequency. So while it goes without saying that zero detectable MRD is what we want, this is kind of a great way, or this is a good way to really assess when we get to those lower frequencies, can we actually detect them? And speaking of detection, in the clinic, detection limits can actually vary, and our concept of clinical complete remission can also be a little bit variable too. And it really comes down to the methods we're using. As we can see from this graph, detection limits vary by these methods. You can see 1 in every 1,000 cells, roughly, for microscopy and FISH, down to 1 in a million, roughly, for next-generation sequencing. And as we kind of push this detection limit down, we can better assess whether relapse is a real potential or is very unlikely. So altogether, by pursuing this use case of minimum residual disease, this aim will really serve to both validate the model and to do it in a real-world, using real-world data in a clinically meaningful way and to a very relevant end. So the data for this aim is 3,210 samples from 698 patients here at OHSU. And of these patients, 236 have 2 or greater samples, or minimum residual disease assays. So this means that we could potentially kind of take a posterior distribution of variants that we picked up from the first assay and use that to inform our calling in the second assay. These samples exhibit variable cellularity, and of the 43, only 41 genes are present, and the sample source is from peripheral blood and bone marrow aspirate. Similar to the fixed dilution series, they've been sequenced on the Ion Torrent PGM platforms using a 43-gene leukemia panel, and the mean read depth is roughly 1651, which is pretty good. This should give us enough power to detect down to a good minor allele frequency. So it's the impact. The validation of current background error models in AIM-1 should provide a meaningful contribution to the understanding of optimal background error models for low-frequency variant calling using GATK and MuTect. And for AIM-2, this project will serve to improve the sensitivity and specificity of MRD assays by the KDL, or the Night Diagnostic Lab, and unlock further potential for agile treatment and improve patient outcomes. In addition to the development of a novel background error model designed for the detection of low-frequency variants, which may be innovative, developing this model with the core tenets of generalizability and extensibility, it should have the potential to augment our ability to detect similarly subtle biological signals across a more broad area of research interests. And this could ultimately yield a lot of benefits to the field of just generally detecting rare biological variation. So with that, I'd like to thank everyone, my committee, and Christina Zhang for her help, the Night Diagnostic Lab for doing these fixed dilutions and providing valuable feedback, Leukemia and Lymphoma Society, which will be funding some of this research, Diane Doctor, couldn't have done this without you, and of course my cohort and the department. Appreciate it very much. Thank you. And questions? Yes. Okay.
So much of this I didn't understand, but it looked great. So the question I'm going to throw at you is a bit of a curveball, perhaps. So what are the ethical implications of detecting minimal residual disease at smaller and smaller levels in terms of impact on patients and healthcare systems?
Ooh, that's a tough one. Yeah, I think I think that's a great question. It kind of gets down to, I would say, boom. I would say that there's definitely some ethical implications of how you provide treatment to patients, whether these assays can actually give them the treatment they need and actually influence what treatments they get, and kind of in how we ration care to these patients. I think that it's definitely, I haven't really quite thought out all the ethical issues personally, so it's definitely something I need to look into more. But I'm sure, in medicine, we're fraught with ethical issues quite frequently, so I'm sure there are some that should be considered very seriously. Thanks. Hi, Nathan.
So you said the current background model that they're using, something with a Q, I forget. BQSR. So what it basically does is correct the base error score. Mm-hmm. And then you said that you were going to look at different models and possibly different places in the pipeline where that would change. So where else in the pipeline do you think that you could introduce an error model, or is correcting background base scores what are the pros and cons of doing that, or some other way?
So some of the error models we've been looking at are actually kind of coming from research into viral population dynamics. And so BQSR is really, it's already done the alignment, and it's essentially updates the quality scores so that downstream the variant caller can do a better job. Some of these error models could be put ahead of it to...
Sorry, so those are alignment quality scores or the base quality scores that come off of the sequencer?
I believe those are alignment quality scores, yeah. So it's already been aligned. So if we kind of look upstream, some of these error models could be used to kind of do essentially better evaluation of the quality of the sequence before we align. And I think really the whole point of that was to say that while BQSR is one approach using quality scores, a lot of these approaches don't actually take a quality score approach. So kind of taking where is the best place to put this error model? Where are we going to violate the fewest assumptions, and what's really, should it be used with BQSR or should it not? One of the error models that I presented, LowFreak, actually encourages the use of BQSR alongside it. So it's not necessarily an either/or. It's really seeking a combination of error models that could work. Does that answer your question?
Yeah, I think it's obviously pretty complicated, and there's probably a lot of different places in the pipeline that you could think to try to improve. But yeah, I also just wanted some clarity on where exactly they were doing this background correction too.
Yeah, it's after alignment, but before variant calling. Yeah. Any other questions? Rick. Yeah.
Well, if you could hold it down.
Hold it down. I'm probably the only non-bioinformaticist in the room, but I guess my question is, what tools are you going to use to assess the various background subtraction methods as to optimability, or whatever word I want to use? How do you know which ones outperform the others? What criteria will you use?
Yeah, so I think that kind of gets to, if I'm understanding your question correctly, I think that's going to be really getting towards the sensitivity and specificity, and kind of when we run the data through GATK, and we run it without BQSR, we run it with BQSR. Say we take model X and we insert it at the most logical point. We're essentially going to look from the baseline of no error model at all, no BQSR or anything, and kind of look at how the variant calls in the truth set match up, and kind of use sensitivity and specificity to see how those align together. Yeah. Does that answer your question? Someone?
It does. I guess the key question being, do you have a truth set? That's what's key. That's what you're going to apply it to. So.
Yeah, and I think the fixed dilution series, there'll be a truth set there, and I think that also with the ICGC-TCGA Dream Challenge data, because it's synthetic, we have a truth set, and we have a known sample purity and known variant allele frequencies. Yeah. So with MRD, I guess I think we also discussed de-identified patient reports potentially in cytogenetics, and those could also offer a truth set there. Right.
With the DREAM data, how rare are the rarest allele frequencies? Do you know?
Not very rare.
Yeah.
I think 18%. Once you account for the purity and how that influences the variant allele frequencies, I think the lowest it gets down to is about 18%.
It's going to be basically, so if you work on that first and be able to detect all those at 18% frequency,
yeah.
Right? You won't really know if you can do better than that, things that are at lower frequency than that. And maybe with the dilution series, you'd be able to get at that, because if you detect something with really low dilution, I guess, so there's a lot of reads there, then you can see, again, if you can detect it when it's much more diluted later.
Yeah,
so.
and I think the reason the reason the TCGA, the synthetic data, the reason it's quite high in variant allele frequencies is because this is whole genome sequencing data. So it's got lower depth, and so our power to detect these is going to be a little bit more compromised when we compare it to the targeted panel, which is what the fixed dilution series is going to be. So these are two kind of different platforms that we're going to be using, and we're going to really see if any models we develop can be used to detect these in both settings.
I guess I was a little confused about that too, sorry, for all the questions. Okay. So in the targeted panel, you're only sequencing certain genes, and there's some targeted pull-down of those genes?
Yeah, so it's a 43-gene leukemia panel, which we've been using here at OHSU. I think now it's moving on, is it 76? Yeah, so now it's a 76-gene panel. So this is historical data.
Okay, so you have something that pulls down just those sequences based on primers that are aligned to those known sequences, and then...
Yeah, I think that would be it. Honestly, I've yet to really get on this, so I can't quite answer that probably satisfactorily right now.
It's a PCR approach.
Yeah, amplicons. Yeah.
Right, okay. So there's going to be some bias there too, right?
Oh, absolutely.
Yeah.
And then you're sequencing them in the same way. They're all in the ion torrent?
Yeah, so both the data for the second DAME and the fixed dilutions in the first DAME are going to be sequenced on the same platform.
Yeah. You know, following up on this question, because my work does recall and precision on document retrieval,
yeah.
and we have challenges with what is a gold standard, and that's what as you were talking, I was thinking of this too. What is the ultimate? What do you consider to be the ultimate truth? When it comes to sequencing, because obviously we can't look at it, the DNA itself, and see what the sequence is, and then I'm always, from an experimental standpoint, nervous about synthetic data. I mean, we use it for a number of things, but you're always wondering in the back of your mind, are there is there something about this data that may be skewing us? So how do you really determine what truth is in these things, and if your determination of what truth is wrong, how robust are your algorithms?
Yeah, so I think that's a really good point. If we don't get truth right, our algorithms are not going to work, really. But I think regarding the truth, we've kind of discussed for the second DAME that cytogenetics represents a pretty good truth. And so that's a totally different, method than like next generation sequencing. So while we can't assess truth, we can hopefully build enough confidence that we can, argue we've kind of pinned down a good level of truth that validates the method. It's slightly squishy, but it should, I think, especially when you get to deeper coverage, you can get a very good idea of what that sequence is, and that's something that we'll definitely leveraging in the fixed dilutions and in the second DAME data. Yeah.
It's just like with judging documents is relevant. There's, it's in the eye of the beholder.
Yeah.
So, but it's good to think about those things. Not that necessarily obviates your algorithms, but it just potentially biases them in one direction or another.
Yeah, absolutely, and that's something we're going to really have to evaluate, like what biases these algorithms may hold, and addressing any limitations. Yeah. Any other questions?
Okay, well. Anyway, since this is a thesis proposal defense, the part after the talk's a little more informal, but your committee will still discuss with you and determine moving on. But thank you very much.
Thank you.